Okay, this is meiosis part 2, and in this video we're going to focus on the stages of meiosis. So if you look at meiosis and the stages that are shown here, the names of the stages you should be able to remember because they're the exact same as the stage names for mitosis. The only difference is we have prophase 1 and prophase 2 because there are two divisions in meiosis. In the first division, um, like in both divisions, things that happen with the spindle fibers and the centrioles, all those things are pretty much identical to what happens in mitosis. So since you've already learned them for mitosis, the big thing to focus on is, okay, what's happening with the chromosomes? That's the main difference of what's happening in these divisions. So in the first division, it's the homologous pairs that get separated. So instead of lining up so that they're all the chromosomes are lined across the plate, they actually pair up and they line up as pairs. This one is actually nicknamed often the reduction division. So meiosis as a whole is nicknamed as the reduction division, but it's this particular one, the first division, that results in reducing the number of chromosomes to half. Okay, so by the end of this first division, we have two daughter cells that are haploid. And so they would have two copies of the chromosome in the X form because they look like sister chromatids still. Okay, because in the next division, they're going to separate the sister chromatids. But when you have an X, what we see as an X-looking chromosome, that is just one chromosome. It's just that you have two copies of it. Okay, so when the homologous pairs are separated, we get half the number of chromosomes in each of the daughter cells. And then in the second division, the sister chromatids will be separated. So we end up with 4 daughter cells, and the cells are haploid with one copy of each chromosome. So in our liver cells, for instance, we would have 2 copies of chromosome number 5, but in our egg or sperm cells that we make, we'd only have 1 copy of chromosome number 5. So this is a diagram that's in your book. I just wanted to highlight a couple of things from it. I wish they had chosen a different way of representing the chromosomes. If you look, they have the homologous pairs in the same color here, and then these two are homologous. They're in the same color as well, where I would rather have seen them have red and blue so that they have a mother copy and a father copy. Okay, so when we go through each of the stages, I'm drawing them in red and blue so you can see the mother-father copies and follow them along in that sense. So if you look very quickly at it, in the start of meiosis, it's the same as the start of mitosis. The cell is in interphase. So the nuclear membrane is intact, and you can't see the chromosomes right now because they're in chromatin form or chromatin form. During that second gap phase in interphase, they're getting ready for division. So now there's 2 sets of centrioles because it's an animal cell. There'd be proteins there ready to make the spindle fibers and all those things getting ready for the cell division to happen. In prophase, the nuclear membrane has disappeared. We now have visible chromatids, they're sister chromatids, and they're going to line up. They're going to find each other, homologous pairs. So in metaphase, you can see them lining up in the middle as homologous pairs. So in the first division, the ones that are pulled apart during anaphase are the homologous copies. And if you notice, you could have had both blue ones on this side or both red ones on this side. It's very random which way they would line up at the metaphase stage. So two cells that result end up with 2 chromosomes each. They started with 4, and now we have 2. So there's half the number of chromosomes. So in the next prophase, there's no, in between the 2 divisions, there's no replication of DNA. So there was only the 1 before any of this happened. So there's no more replication of DNA. So we start off with those 2 chromosomes again that we had finished off in the first division. They're going to line up. Looks a lot like mitosis, because then the sister chromatids are all pulled to opposite plates, and we end up with opposite poles, sorry. And we end up with 1, 2, 3, 4 cells, each of them with 2 chromosomes. So that's the end of anaphase. At the end of telophase and cytokinesis, there are 4 separate cells. Each of them are haploid. So we're going to take a look at the stages. Actually, I'll show you the animation first, and then we'll take a look at the stages in more detail. So hopefully this will show up okay. There's a link for it on the class notes page too, under links. If it doesn't happen to show up as well, you can watch it there. I'm just going to start that again because I don't know, some silly little thing is interfering. Okay, so we're going to take a closer look at each stage. If you remember, an interphase is pretty much the same as what we saw during mitosis. I'm just going to make this bigger, and I'll get a pen. Oh. Okay, so if you look at what happens during interphase, it's the same as what happens during interphase before mitosis. We have gap 1, where there's growth synthesis stage where DNA is replicated, gap 2, where it's getting ready for division, so there's copies of centrioles made, there's new proteins, enzymes, all things getting ready for cell division. DNA is not visible through this phase because it's in chromatin form, or chromatin form. In meiosis 1, the reduction division, we start off with the diploid number of chromosomes, and we're going to end up with half, right? So during prophase, if you notice, centrioles are migrating to the opposite poles. You can see the sister chromatids. They look like X's. Okay, the membrane starts to dissolve. Um, they become visible, same as during mitosis. What's different in this case is the homologous chromosomes pair up in a process called synapsis in order to form what we call tetrads. Tetrads, because there's four copies, two in each chromosome, because there's a sister copy of each, so we say it's a tetrad. Okay, so they form tetrads. And when that happens, if you notice in the animation, we can have what's called crossing over, where little bits, where the two arms of the homologous pairs cross over each other, they can actually exchange material. And so what you get are all different sister chromatids. Okay, so even though in mitosis, the sister chromatids stay exactly the same, genetically identical, in meiosis, the sister chromatids can be different because of crossing over. So that's one source of genetic variation. We're going to talk about that a little bit more later as well. In metaphase, just like in mitosis, the centrioles are now at opposite poles. Spindle fibers the spindle's complete. The spindle fibers attached to centromeres. But in this case, the homologous pairs line up at the equator. So the mother-father copies line up, and it's very random. This is another source of variation. You could have all the father copies on one side and all the mother copies on the other, or you can have um, the opposite. So we could have a mother copy on one side and a father copy on the other, and then the father copy of the other one beside it, and the mother copy on the opposite. So it's very random how these can be. And if you think if we have 46 pairs, or sorry, 23 pairs, so we'd have 23 randomly organized on different sides of the equator. Okay, so that's another source of genetic variation. The spindle fibers are going to attach to the tetrads at the centromeres, but when we get to anaphase, the sister chromatids aren't pulled apart like they are in mitosis. Instead, the spindle fibers are going to pull the pairs apart. Okay, so they're going to pull the mother copy to one side and the father copy of the chromosome to the other. Can you see? I don't know if you can see it very well, but these ones have the other color at the tips of them because of the crossing over. See that? Okay, so if I were to look at the two copies, these two sister chromatids, then they're not both all red. One of them has a bit of blue on the end because it's exchanged some genetic information. So that's what happens in crossing over. There can be an exchange of genetic information, which means in the end, we end up with very unique gametes. So during anaphase, the homologous pairs are going to move to the opposite poles. The sister chromatids are still attached to each other. Okay, so when we reach the end of telophase and cytokinesis, where the cell pinches inward, forming furrow, and eventually the cells divide completely, we end up with the nucleus has reformed. Really, you wouldn't see these, but I'm showing them just so you can see that they have all four different versions, and we can have a random assortment of mother-father copies. Okay, I just wanted you to see what the chromosomes look like. So normally they would be in chromatin form at the end, okay, but I'm just showing them so you can see. So we end up with two daughter cells, and each of them with their own nucleus, and half the number of chromosomes. So this is where they are haploid already. The number of chromosomes started with 4, and now there's 2. So there may or may not be a short interphase between the two meiotic divisions, but whether or not there is no further DNA replication. There was only one time that DNA was replicated, and that was before the whole process began, during the S phase of interphase. So in the second meiotic division, prophase, again, the nuclear membrane starts to dissolve, the chromosomes become visible, the centrioles start to migrate, so they're um, at opposite poles. Now, they're going to migrate so they're kind of perpendicular, or at 90 degrees, from where they were at the first division, and that's just because of positioning. And that's not really a thing of great importance to remember. Okay, it's just the way it tends to happen. Spindle fibers begin to form. Okay, but really it looks a lot like mitosis, except they are still sister chromatids, so they still look like X's. Whereas in I don't know what I was going to say. In normal mitosis, no, never mind. Okay, by the time we get to metaphase, then these guys are going to line up in the equator. So we have a mother copy and or a father copy of, well, not both for that one chromosome, but we have one copy of each chromosome that are going to line up at the metaphase plate, okay, in the middle. So they're still sister chromatids. Here it looks a lot like mitosis, but it's different from mitosis in that at mitosis you would have the original number of chromosomes. Okay, so if you started with 4, then there should be 4 lining up here. But since there's only 2, then we must have already had a division that made them half the number of chromosomes. Okay, so at this metaphase, the chromatids are lining up and the centromere is attached to them, a lot like in mitosis. A little slow to respond. So in anaphase then, the centromere is split and the spindle fibers are going to shorten, pulling the sister copies to opposite poles. Okay, we can pull the sister copies to opposite poles, and of course they look a little U-shaped, although not so much in this diagram. In telophase, the sister chromatids are now at opposite poles. The nuclear membrane is going to reform just like in mitosis. The furrow starts to happen. Sometimes it happens at the end of anaphase, but basically cytokinesis is also happening at the same time. And again, I haven't shown it in chromatin form here just because I wanted you to see that we've gone from having 4 chromosomes to 2 chromosomes. It's already been halved by the end of the first division. Okay, but each of these cells that's made has a unique combination of the chromosomes. Okay, so if you look, no 2 of them have a small all blue chromosome. Okay, this one has an all blue one, but this one has a small blue one, but it has a little red tip. Okay, and no 2 have the same combination of entirely same combination of genes. So that's why, depending on what egg and sperm you get from your mom and dad, you might look similar to your siblings, or you might look very different. You might look similar to your parents, or you might look very different. Okay? So, during cytokinesis, cytoplasm will divide to create 2 daughter cells. So, by the end of meiosis, we have a total of 4 cells. Okay, 2 by the end of the first division, and then those 2 divide again, so we end up with 4. Each cell has its own nucleus, nucleolus, like normal, but each is haploid and not identical. Okay, they're all unique. So, we talked a little bit as we talked about the stages, about the sources of genetic variation. And this is really, really, really important. So not only is it important that meiosis reduces the number of chromosomes to half, but also it allows for genetic variation, which is important for natural selection, um, so that we're not all the same, basically. Okay, so we end up with a random assortment of the chromosomes in the sex cells and the gametes, but for really for a couple of reasons. So we have random assortment, the way they line up at metaphase. Okay, so because they can line up randomly at metaphase, then it's very random whether we get mother or father copies of certain chromosomes. The other source is through crossing over, where regions of the chromosomes, the homologous pairs, are exchanged during synapsis, during prophase, while they're stuck together. Okay, so this diagram is showing the tetrad, and the tetrad where certain regions of the homologous sister copies are crossing over and they can exchange genetic information. Okay, so if you look here, these are different. This one is big A, big B. This one should be big A, big B if they were identical copies anymore, but they're not. Same as these two. They're not the same anymore. So it allows for very different combination of traits. Okay, this one is showing that we're going to watch a little animation of it actually. So I need the other. And this animation again is linked from the class class notes page. So if it doesn't show up very well, then please watch it from there. My computer's a little slow, sorry. All right. Oh, what are you doing over there? Hide this. There we go. Ignore the ads. I don't know why they're there. Here we go. Just thought I'd pause it so you can see what they mean. Okay, if you look at the left side versus the right side, oh, alarm clock going off. Sorry. If you look at the left side versus the right side, and each of those is representing a different chromosome pair. So even though they look similar, it's the ones on the left and right. The purple and green copies are the same. I need a pen. Where's my pen? Okay, so for instance, these would be chromosome 1. These would be chromosome 2, and these would be chromosome 3. And let's say the dark color is the father copy and the light color is the mother copy. So in this one, all the mother copies are on the left, all the father copies are on the right. In this one, all the father copies are on the left and all the mother copies are on the right. And in this one, we have a mixture, okay? And this is a different mixture, even though only one father copy is on the left, it's a different chromosome. So this is chromosome 3, but this is chromosome 1. So it's very random. And if we think of humans, then we would have the possibility of 2 to the power of 23 possible combinations. So that's a lot of different combinations when you think about it. Hopefully that showed up okay. Okay, so random assortment. That's one major source of genetic variation. The other is the crossing over. So we'll take a look at the animation of that. Maybe the one that we watched earlier, but oh, couldn't remember. It's going to move again, so I'll just wait till it's done. There we go. All right, so I'm sure in your assignment you've seen questions that relate to these two things, so hopefully you understand them a little bit more now. If you have any questions, please email or text me, and I'll gladly help you out. So that's meiosis, the stages, and sources of genetic variation. The next video will deal with, I believe we're going to talk about meiotic non-disjunction and disorders that happen when that happens, when meiosis goes wrong.